welcome back to the course of plant developmental biology. In today's class, we are going to discuss about root development. So, as we discussed in some of the previous class, that if you take a mature plants, this can be divided into two system. One is the root system and shoot system. Root system is usually below the ground part of the plant. Shoot system is above the ground part of the plant. And if you look a closer view of the very tip of the plant at shoot, this is a scanning electron micrograph picture of growing shoot apical meristem, which is basically inflorescence meristem. And at the very tip of the root, this structure is called root apical meristem, which we will look in detail. So, this slide also you have seen in one of the previous class. So, if we take a general root development in plant, so there are three major developmental processes which is going on during the development. First process, which is called root apical meristem maintenance, which occurs at the very tip of the growing primary roots. This is a typical primary roots in model dicot plant. And if you look at the very tip, this is the region which is called root apical meristem, and this is the region where some cells retain their identity as a stem cells. Stem cells means they can only undergo the process of cell division, they do not enter in the process of cell differentiation, and this is very important for continuous growth of the root. For development of the root, for growth of the root, or for lot of organogenesis, always a continuous production of cells are required, and this is happening here in the root apical meristem. And then if you come slightly later, zone of or slightly upper zone of the root, this zone is called zone of cell expansion, and in this zone mostly tissue patterning, the second part of the development in the root takes place, where different tissues or different layers of the cells, they take a very special identity. And then if you come in the even higher up region of the root, the process of proper differentiation of new organs or organogenesis occurs, which is mostly in case of root is lateral roots or root branching. So, the process of root branching is visually initiated in the differentiation zone, and a lot of lateral roots are developed in this region. So, now in this class we will focus on the first aspect, and which is root apical meristem. What happens in the root apical meristem, what are the process, how the meristem is maintained. This is more important, because if you look the root tip carefully, so in this picture you can see this is the region which is basically the meristematic region. Meristematic region is the region which undergo the process of cell division used mostly, and this is the region of cell elongation, and then here is the differentiation. And if you take a closer view, this is how root tips looks, and if you make a cross section, so you can clearly see that these are the different cell layers. The outermost layer is epidermis, then you have a layer of cortex, then you have endodermis, then this is pericycle. In pericycle there is a xylem axis, so this is protoxylem, this is metaxylem, and then at two poles you have phloem poles where you have sieve elements as well as companion cells, and then you have some procambium cells. This is a longitudinal view, so you can see this is epidermis, cortex, endodermis, pericycle, and the vascular tissues. But apart from that, you have some tissues here which is called columella cells, and these are lateral root cap. So, all these are part of a particular growing roots. How this process occurs? So, the first thing is that how the apical meristem is established, how this organization or proper organization of the root meristem takes place, and what is the importance of all these cells. And then this happens at a very early stage of the embryogenesis, during the embryogenesis. So, if you again recall one of our previous class where we have discussed that the first diploid cells after fertilization is zygote. Zygote undergo the process of embryogenesis, where it undergo several round of mitotic cell division, And at some stage, for example, if you look at late globular stage, you can see there are three domains, three distinct domain. This domain is eventually the upper domain or apical domain, which is eventually going to make the upper part of the plant. Then you have a middle domain here, this will make eventually the hypocotyl region of the seedling, and this lower domain is basically going to make the root. So, important thing here is that the program, the basic developmental program for root shoot is established at the stage of embryogenesis itself. Here you can clearly see that this is the region of the root apical meristem, which is already established. Now, if you look the growing root, once it is immersed, and you can see this tip, this is the very closer and high magnification view. So, few cells which you have to understand very early to understand the entire process of root development. So, if you look, this is the root tip, and this red color cell, this is very important cell. So, you can see here 2, but actually they are 4 in numbers. These cells are called quiescent center, this is QC, and the feature of these cells are that they themselves are very slowly dividing or almost not dividing, but they regulates the division capability of the cells which are close to them. So, if you look this boundary domain, this is the region which is called stem cell niche here, because the cells which are lying in these domains, they will not undergo the process of cell differentiation, they will only divide, they are the stem cells. These are also called in plant initial cells. And one important thing here is that, and you will also realize later on that all these initial cells are the stem cells, they are directly connected with the QC, which means that the cells which are directly connected with the QC, they might be receiving some kind of signals which basically allow these cells to remain as a stem cells or the cells which are dividing, and it also helps them not undergoing the process of differentiation. Then apart from that, you can say these cells depending on their position, these initials, these are like steel initials, and if you look here, this is initial, and actually for cortex and endodermis you have a common initial which is called CEI, which is cortex endodermis initial, but for epidermis, and these initials are basically, if you look, this the D1 layer which is just below the QC, these are essential for columella cells. So, this entire region is columella. In columella, this immediate down to the QC, they are the initials, they are the stem cells, columella stem cells, but other cells which are in D2 layer, D3 layer and D4 layer, they have undergone the process of differentiation, columella specific differentiation. And these cells are basically making lateral root caps. Another important thing which is important here to know that these columella cells, when they undergo the process of cell differentiation, they accumulate starch, and there are way to detect the starch. So, if you detect the starch, if you see the starch accumulation in the cell, which is a marker that this cells has undergone the process of differentiation, these cells are no longer working as a stem cells. And this is important, and then these different layers you can see clearly: epidermis layer, cortex layer, endodermis layer and steel layers. They are arranged in a radial manner. So, next the thing was that there was report when it was studied that what is the importance of QC, because QC looks like that it is sitting in a very central position and it is regulating stem cell property, and what is actually its importance, how it is doing this function. And one very specific experiment was done. If you again recall, in one of the class we have discussed about the experiment which is called laser ablation, through which you can specifically kill a particular cell in the growing root, and what happens if you kill the QC. So, for example, this is a typical same structure which you have seen in the previous slide. So, if you look this picture, so this is your QC, one of the QC is here, but second QC which is now very small here, this QC is ablated. And if you ablate the QC and look what happens to the cells which are in contact with this QC, you see here this is a starch granules accumulation, just now I say. So, if you see this starch granule accumulation, which means that these cells are not stem cells, but these cells are undergoing the differentiation. But if you look, this is the QC, and just below the QC, the cells which is in D1 layer, they do not accumulate the starch, which means that they are basically stem cells. But the cell layers below the D1 like D2, D3, D4, they start accumulating the starch, which means that they have lost the stem cell property and they have started cell differentiation. But if you look this case where one of the QC was depleted, the QC which is intact, the layer which is below the QC, or the cell which is below the QC in the D1 layer, or which is called columella stem cells, this is still maintaining the stem cell identity. It has not accumulated starch, but the cell which is below the ablated QC, this has started accumulating starch, which means that this cell has lost its capability of stem cells and it has started differentiation. So, which tells that QC is very, very important in maintaining a cell as a stem cell identity. How this is possible? What could be the hypothesis? In first hypothesis, what can happen, that QC is activating division and repressing differentiation simultaneously, independently. In second possibility is that QC is actually activating division and the result is inhibition of differentiation. In third possibility, QC is actually repressing differentiation and result is activation of cell division. But if you look this experiment, if you look this data, this suggests that the third pathway is working. Then on the basis of this, one model has been proposed, and the model here is that there are basically two types of signals. One signals which might be coming from the very mature cells of the upper region of the root, and they are the differentiation signal, they are promoting the differentiation. And another signal which is coming from the QC, going out from the QC, and this signal is basically inhibition of differentiation. So, basically both the balance between these two signals might be important. The cells, and this signal is more kind of short distance signal. So, the cells which are in direct contact with the QC, they are receiving this differentiation inhibition signals, whereas these signals are coming from top. And what happens, this and here, if you have inhibition signal, the differentiation is inhibited here. But once these cells divide, so let us assume if this is an initial cell, and if this cell divide, the cell which is present at the upper side, now it loses the contact with the QC, which means that it might not be getting the signal, the inhibition signal, or the signal which is responsible for differentiation inhibition, but at the same time it is getting signal from the top, which is to promote the differentiation. Now these cells will start the process of differentiation, whereas the lower cell which still remains in contact with the QC, it will retain the process of cell division, and process of cell differentiation is inhibited in these cells. So, the next question comes that okay, QC is important for maintaining cell differentiation, balance between cell division and cell differentiation. This is crucial. What could be the genetic pathway? What are the regulators, and what are the regulators, and how they helps in positioning this stem cell niche in the root apical meristem? And one of the pathway is scarecrow short root protein mediated pathway. So, scarecrow and short root, these are two grass family of transcription factor. They are a special transcription factor and they are expressed in a tissue specific manner. So, here if you look the expression pattern of scarecrow, promoter activity of scarecrow driving GFP expression. So, it expresses very well in the QC and the initials for cortex and endodermis, but at the later stage the expression is only restricted to the endodermis and there is no expression in the cortex. But in scarecrow mutant, what happens that the expression in QC disappears, this reason. Similarly, if you look other markers, these are the QC25 and QC46, they are the marker genes which are known to express in the QC. But in scarecrow mutant background, you can see that the expression is totally lost, which means that in the scarecrow mutant, QC identity is defective, which tells that the identity of QC is regulated by scarecrow. But this is not always. Some of the QC markers, they still get expressed in the QC. So, maybe the identity is not completely lost, or maybe this could be the position dependent expression of the genes. But another interesting thing, if you look here, so this is wild, if you look here, the granules accumulation, starch granule accumulation. So, if you look in the wild type, this is the QC, and this is the columella stem cells, there is no granule accumulation, but in absence of scarecrow protein, the cells just below the QC, they start accumulating starch. So, this tells that the stem cell maintenance, columella stem cell maintenance is defective in case of scarecrow. This can also be seen here. So, this Q1630 is marker for differentiated columella cells. So, as you can see in the wild type case, this is QC, and this layer, the D1 layer, which has columella stem cells, this gene is not expressed here, but the D2 layers, which has the differentiated columella cells, express Q1630. But in scarecrow mutant background, you can see that the cells which is just below the QC can start accumulating columella specific markers. This all together suggests that scarecrow is important in not only taking QC as a proper identity, but also positioning stem cell initial at the right position. Another important pathway or genetic regulation of positioning root stem cell niche is mediated by another class of transcription factor, which is plethora. Plethoras are AP2 domain containing plant specific transcription factor, and this is very important. And if you recall one of the class, we have discussed that usually they work in a genetically redundant manner, and that you can see here as well. So, when you have a single mutants, you do not see a very strong phenotype, but when you combine plethora 1 and plethora 2 double mutant, you can see that root growth is inhibited. So, it is a very short root plant. And if you look the differentiation pattern, so this is the QC, again the same thing, but if you look the double mutant, the cells just below the QC in the D1 layer, it has started accumulating starch, which suggests that in double mutant plethora 1 and 2 double mutants, the stem cell niche is not getting maintained. Stem cells are losing their stem cell property, and this is also visible from here. If you look here, this is double mutant plethora 1 and 2 double mutant, this is wild type, and this is the basically size of meristem. So, in wild type, this is the meristem size, and this meristem size is significantly reduced in the double mutant. Here, if you look the marker, cyclin Gus marker, cyclin markers are basically markers which marks the dividing cells. It will express in the cells which is under process of cell division. And if you look in the wild type, you can clearly see that large number of cells are under the process of cell division, but in double mutant this number is significantly reduced. Same thing you can look by lack of the QC25 markers. So, in wild type you have QC25 markers expressed in the QC, but in mutant the expression is totally disappeared. But then the question is that we know that there is already regulated scarecrow and short root pathways regulating positioning of the root stem cells. Now, we are coming that plethoras are also regulating. Are they working through the same pathway, or they are working independently? So, to check that, if you look these mutants, so basically this is the double mutant, and if you look the expression pattern of short root scarecrow protein and short root protein, and what you find that the expression looks quite normal. So, in the embryo stage between wild type and mutant, here also wild type and mutant. So, the expression pattern of both short root and scarecrow protein is not significantly affected in plethora mutants. This suggests that they might be working independently for regulating a stem cell niche positioning in case of root apical meristem. Okay, so if we combine here, so what you can see here, that there are two pathways, short root scarecrow pathway and plethora's pathway. Short root scarecrow pathway, basically they are helping in positioning QC in radial manner, whereas so radial manner is basically in this manner. So if you look, this is the QC, so they are expressed here, short root proteins are. So if you look this picture, this is the expression pattern of the genes. So this MP we will talk maybe later on. This is a kind of genes which is regulated by auxin, and auxin is very important hormone which plays a very important role in the root development. But apart from that, if you look the plethora expression, plethoras are expressed here, this domain. But at the later stage, if you look, or maybe in the growing meristem, if you look, so this is the cells where plethoras are expressed. This is the cell where plethoras are expressed. This is the cell where your scarecrow and short root proteins are expressed. But if you look this cell, so this is basically QC as well as this CEI initials. So cortex endodermis initials. These are the cells where all three genes are expressed: plethora, scarecrow, and short root. So basically, the hypothesis is that short root and scarecrow plethoras are helping in positioning the stem cell niche in the radial manner, whereas plethoras might be helping in positioning the stem cell niche in the longitudinal manner. And this is very important. So later on we will see that actually short root activate expression of scarecrow and it activates expression of Vox pipe, which we will see in another slides. So here there are two pathways which are promoting stem cell niche formation and helping them to position in a right domain. But there are some repressors of this process, and those repressors need to be repressed to ensure that a proper stem cells are maintained and proper stem cell niches are positions. And one of them belongs to another class of transcription factor which is called homeobox transcription factor. And this you can see here that if you look the mutant of serrate gene, serrate is a nuclear zinc finger protein. In this mutant, what you observe that the proper embryogenesis is totally defective, and there is no proper organization of the cells, there is no proper organization of the apical meristems. But what happens that if you combine this mutant, serrate mutant, with some of the homeobox genes like fabulosa and fabulota, these are the homologous genes. If you make double mutant or triple mutant, what happens that this defect of serrate mutants is complemented. What does it mean? It means that somehow they are genetically interacting and they are basically suppressing the phenotype of serrate mutant. How this happens? So if you look the expression pattern of this Vox homeobox gene fabulosa, so you can clearly see this is very asymmetrically expressed or localized. You can see it is only present in the apical domain, it is not present in the basal domain or root apical meristem domain. This is important. So this gene has to be repressed in the root apical meristem for proper meristem function. But what happens in the serrate mutant? In serrate mutants, this asymmetric expression pattern or transcript localization is lost for the fabulosa genes. Now you can see the fabulosa is expressed everywhere in the embryo, and that is disturbing the pattern, that is not allowing a proper root apical meristem to take place. But when you combine this, when you knock out this transcription factor in serrate mutant background, early there might be not much complementation, but later stages this basically complement the phenotype. As you can see through the marker, so this is a scarecrow marker. Scarecrow, just now you have seen that it expressed in the QC as well as the endodermis and initials. This is Vox5. We will more talk in detail in the next slides, but here just to know, mark here that this Vox5 is another homeobox protein, but which express in a very specifically in the QC cells. But what happens in the serate mutant, both the markers lost their expression pattern, which means that there is no proper QC identity, there is no proper stem cell niche organization and positioning. But when you combine, when you make a double mutant, triple mutants, what you can see that they reappear, their expression domain reappear in a right domain, which means that now if you mutate Fabulota, they basically this mutant, the serate mutant or the triple mutant, they can reorganize their stem cell niche, root stem cell niche, and that is why they can rescue the phenotype. So, if you look this model, this Fabulosa protein which is which is highly expressed in the apical domain and it is kept repressed in the basal domain. At this stage, particularly in root apical meristem, this needs to be totally kept repressed. So, there are two kind of mechanism. So, if you want to maintain root apical meristem, then some activator has to be activated, at the same time the repressor has to be negatively regulated to ensure proper development. Another important regulatory pathway which is regulating root apical meristem maintenance is WOX-CLE ACR4 module. So, we will go one by one what are the interaction. If you recall our previous few classes where we have discussed in detail what kind of approaches we are taking to study. Now, here what you are going to learn that how those approaches has been used and how the data has been interpreted to understand a particular developmental pathway. So, this WOX5 is another homeobox domain containing transcription factor, but it has more positive role on the stem cell, root stem cell maintenance or QC identity, unlike the Fabulosa and Revolota. If you look the expression pattern, so this is WOX5 promoter activity or RNA, you can clearly see that it is very specifically expressed in the QC. And if you look, this is wild type, so this is QC, just below the QC you have columella stem cells and then columella, but if you look in the WOX5 mutant background, this is the QC, but the cell just below the QC it looks defective, it is not normal cells. And if you check the markers, different markers of the QC, for example QC 184, you can clearly see that though QC identity is not completely lost here, there might be maybe some of the markers are disappearing, for example QC 184 is disappearing, but WOX5 marker is still active. So, there is a defect in the identity, but another important thing, if you look here in the wild type, this columella stem cells identity is basically here lost and it has become more kind of differentiated. So, this has accumulated the starch granules, which tells that WOX5 is important for stem cell maintenance, for columella stem cell maintenance in the root. Then there is another peptides which is called CLE peptides, CLE40 genes. If you look this mutant, so this is wild type. In wild type typically you have QC, then you have one layer of D1 layer which is your columella stem cells, and then next layer is your columella cell, differentiated columella cells. But if you mutate CLE40 genes, what you see that there is more than one layer of stem cells, columella stem cells, which means that in CLE40 mutants there is a gain of stem cell activity. So, there is more than the required stem cell activity. And but if you take this wild type and treat exogenously with CLE peptide you can synthesize, and then what happens that even the one layer of initial cells or stem cells has disappeared in wild type. So, in wild type there is a endogenous CLE40 signaling is active. Now, you are putting more CLE40 protein from exogenously. So, it might be working in the dose dependent manner, and if you have more doses, even one layer of this activity is lost. But if you look here, when the endogenous CLE40 pathway is lost because of the CLE40 mutant, now you are supplementing CLE40 from outside, it basically restores the phenotype to the wild type. Here you can see only one layer of columella stem cells, and these are just marker expression pattern for WOX5 RNA and QC 184 RNA. So, in this way you can say that WOX5 is a activator or positive regulator of stem cell activity, whereas Cl40 might be negatively regulating the activity, but we will see how exactly this is happening. Then if you combine this mutant, if you look the WOX5 mutants, as you see earlier, in WOX5 mutant you do not have columella stem cells, but when you treat with Cl40 proteins, even the QC got differentiated. So, apart from the columella loss of columella stem cells, even QC has accumulated the gain. So, there is a gain of differentiation. Then there was another clavata like genes, which is clavata 2. Maybe we will see in the shoot, when we will discuss in the shoot, the similar kind of mechanism functions in maintaining shoot apical meristem. So, if you look clavata 2 mutants basically it does not have any phenotype, it looks very similar to the wild type Columbia. Then if you go again and look another mutants, or if you see the genetic interaction with other genes, then what you can see, there is another clavata which is called clavata 1. In clavata 1 mutant, this shows a phenotype which is very similar to loss of clavata 40, and another gene which is called ACR4, which is basically Arabidopsis crinkly 4. So, this also phenocopy clavata 40 mutant phenotype, which means that, and another important thing, if you treat these mutants with the clavata, basically in clavata 1 the phenotype is lost, in fact it is more differentiation has started, but if you look in ACR4 mutant, even after clavata 40 treatment there is 2 layer of columella stem cells. This suggests that they are working through the same pathway, and clavata 40 is upstream of ACR4. So, it is basically clavata 40 is working through the ACR4. So, if you look here, this is Cl40 and then downstream is ACR4. So, if you do not have ACR4, even if you provide clavata 40, the phenotype cannot be rescued. So, ACR4 is required for clavata 40 to work here. Then if you look again, these are the double mutant phenotype. So, this is Cl40 alone, there is 2 layers. When you combine clavata 1 and ACR4, there is 2 layer, clavata 1 and Cl40 2 layers, clavata 4 and Cl40 2 layers. So, all are having similar phenotype, which suggests that all of these might be working through the same pathway, same genetic pathway. And then if you see the expression pattern of them, you can find this way. So, this green is ACR4 expression pattern, and red is clavata 1 expression pattern, and this is the Cl40 expression pattern domain. And another important thing what has been seen, that this ACR4 is very specifically localized to the plasmodesmata. Plasmodesmata, in some of the future class we will see that plasmodesmata is a channel through which lot of cell to cell communication takes place during the plant growth and development. So, here if we combine all these information together, so what you can suggest here is that clavata 40 which is present here, this clavata 40 is actually the biochemical nature of clavata 40 is known. This is a kind of signaling peptide which works as a ligand, and then these are the receptors. So, this clavata 40 is received by ACR4 and clavata 1, and then this signaling is very important to restrict the domain of WOX5 to the QC. So, QC should not expand, and this is very important to ensure that a position of QC has to be fixed at a particular place, and once you fix the position of QC, eventually you are going to regulate or positioning of entire stem cell niche. So, if you look here carefully, so there are multiple pathway, plethora mediated pathway, short root scarecrow mediated pathways, and then WOX5 ACR4 Cl40 mediated pathway, all of them working together to ensure proper root apical meristem maintenance and the function. Apart from these regulators, plant hormones which are very important in growth and development, almost they regulate every aspects of growth and development. auxin, cytokinin, gibberellic acid, all these hormones are very, very important. So, here if you look, we may not go in a very, very detail because this is a very complex process of hormonal signaling, but something to say that auxin, brassinosteroid, all these hormones are also playing very, very important role either directly or through the same genetic regulatory pathway which just now we have discussed to ensure the proper root apical meristem maintenance. For example, if you look here, this is scarecrow. Scarecrow is negatively regulated by or repressed by the cytokinin. This is response regulators which works in the cytokinin response processes, and then this is auxin. Auxin is activating some of the auxin response factors, and these auxin response factors are regulating the activity of WOX5. Auxin is also regulating the activity of plethoras. So, basically if you look in this picture, you can see that there is a crosstalk between auxin and cytokinin together, which ensures a proper root apical meristem and proper meristematic activity and a critical balance between cell division and cell differentiation during growth and development. And then this is a very complex picture, we will not go in the detail, just to highlight something that apart from auxin and cytokinin, ABA, JA, brassinosteroid, all other hormones are having role and functions in root apical meristem maintenance. Then coming to the another very interesting thing here is control of QC. So, basically QC, just now I said that QC is very, very important for maintaining root apical meristem activity, but QC itself does not divide very actively. So, could it be just that its function is only to regulate the activity, or can it have more function? So, under normal condition it is only regulating this, but in some stress condition it has more functions. So, basically it can work as a reservoir for providing stem cells basically. So, for example, if you look, if there is a damage of the growing root, or if due to some reason if these initial cells or the stem cells are damaged or they are dead, then under that condition this QC basically start dividing. So, there is a activation of cell division in the QC which is normally silent, and this QC basically recovers entire stem cell niche here, so that root continue growing, and this will basically leads to the recovered state of the root apical meristem. Another thing you can see, these pathways are also tightly regulated genetically, but in normal condition if you look, stem cell niche is basically maintained by this QC, but at this stage QC is not dividing. But if there is some stress induced signal or any issues, then what happens that this QC basically starts a process of cell division just to generate more stem cells, and these processes are regulated by multiple pathway, WOX5 mediated pathway, cytokinin, RBR scarecrow mediated pathway, and some of the other pathways, but brassinosteroid and ethylene hormones are also playing important role. So, we will stop this class here. In next class we will move towards the balance between cell division and differentiation, and particularly vascular tissue development. Thank you very much.